For those who are just joining, I'm going to give just one more minute, let a few more people log on, and then we'll begin our webinar shortly. All right. Welcome everybody to the fourth in this series of webinars that FreeThink Technologies is sponsoring. The webinars that we have already had included Maria Ultramari, who presented on new approaches to set acceptable intake levels, Rachel Hemingway, who presented on in silico tools, Steve Baertschi, who presented on the chemistry, and today's webinar, which will be presented by Gary Scrivens, and I'll do an introduction to him and his topic in a minute. This today will be on understanding and reducing nitrosamine formation in solid drug products. For those who don't know me, if there are those like that, I'm Ken Waterman, and I have a cartoon version of myself, which is right here. So just to tell you a little bit about FreeThink, since we're sponsoring this, we like to think we are free, we think free and try to achieve more that way. We're a very science-driven company, and we try to use world-class science to speed better products to market. We're well known for our software, ASAP Prime, but also our laboratories that determine shelf life and try to solve stability problems. Many people might not realize we also have quite a drug delivery technology group and do work, including our latest introduction of a new technology, which is an oral osmotic controlled release capsule called OsmoCap. And we do work in many more challenging areas like depots and solubilization technologies. Let me tell you a little bit about Gary. I've known Gary for quite some time, and Gary is kind of the scientist doing the kind of scientific work that many of us always wish we had time, energy to do, and he publishes quite widely. He is currently, at least for a little while longer, an associate research fellow at Pfizer. As he let us know this morning, though, Pfizer will be closing his site. So anybody looking to hire may be able to make a great offer to Gary. Gary got his bachelor's and doctorates in chemistry, doing his doctoral work on magnetic resonance, ESR and NMR, for mechanisms and kinetics of organic oxidation reactions. He has greater than 25 years experience, which is starting to make him sound old, in analytical chemistry, mostly in the pharmaceutical industry, working in areas ranging from inhalation to dissolution. He's very well known in the stability arena, developed, refined, and applied accelerated studies to the prediction of long-term, but not just applying things. He's really worked to understand the root causes of poor stability, and he's been involved in troubleshooting pharmaceutical product problems to improve product design. He's authored numerous regulatory submissions and more than 30 scientific papers, and also co-authored a book on accelerated predictive stability. So today he's going to be talking about nitrosamine formation in solid drug products. Before I turn it over to Gary, I want everybody to note that at any time during or after, you can type in questions. I would suggest if you're typing in a question in the Q&A section, that you please, if it's referring to a particular slide, you can refer to the slide number. I will work to read off some of the questions. I probably won't get to all of them. Any questions that were not answered, we will forward to Gary, who will try to answer them separately, or if they're addressed to FreeThink, we'll work on those as well. The slides and recording of this and past presentations will be available shortly, and those will also have closed captioning, and that can be found at the link indicated. I also want to put a little plug in for our Science of Stability Conference, which should be, as it's always been, a very good scientific conference. This time it'll be 13th and 14th of May, 2024 in Barcelona, and you can find information on that and registration at stabilityconference.com. And with that, I will turn things over to Gary.
Thank you very much. Yes, I'm just going to share my screen. Hopefully the audio is good, and I will start any moment now. So hopefully the audio is good, and hopefully you can now see my slide, my opening slide. And if
Good.
Yeah, good, thank you. And I'll put it in presentation mode. Hopefully things will still continue to work properly. Okay, so yeah, it is a genuine pleasure to be part of FreeThink's webinar on webinar series on nitrosamines. Yeah, it's come at a very interesting time for me personally, as Ken has explained, but better not dwell too much on that at the moment. Yeah, I'm very pleased to be part of this series. This final sort of slide deck is on nitrosamine formation in solid drug products, and I think the emphasis here is solid drug products. So without further ado, I will carry on. So I've got a couple of slides of introduction. I think this will have been covered in previous webinars, but just in case people didn't attend those, you know, nitrosamines are a very big issue that faced the pharmaceutical industry, starting back in 2018 with the observation of nitrosamines in sartans. There have been many recalls of products across the industry. And the other revelation really is that nitrites are found in many excipients, even though it's not listed on the C of A. And the ppm levels that are present in excipient is enough to cause an issue, because the safety threshold for nitrosamines has been set extremely low historically. I know there's been some development on that front, and there is more of a phased threshold now based on structure, which I think is very welcome in the industry. But nevertheless, the nitrosamine limits are still very, very challenging to meet those levels. And the purposes of this presentation really is to somewhat focus the attention onto solids, as I said earlier, because much of our understanding about nitrosamines has really been from the solution chemistry. And I think much less is known about the formation in solids. Okay, and so the purpose of this presentation is to focus on solids, and it is very much a focus on the science as opposed to any sort of regulatory developments, etc., which will be covered in other presentations. So without further ado, going on to the mechanism. So this is really from solution-based understanding. So first of all, we have sort of the overall stoichiometric reaction equation there. It's effectively the addition of NO, NO plus, and the loss of H plus. So that's the overall stoichiometric equation we're talking about here. And as you can see, it's about specifically secondary amines reacting with nitrite to form nitrosamines. Hopefully that's all. People have got a fairly reasonable background level of understanding on that. And yeah, just to talk specifically about the actual nitrosamine nitrosating species, it is a slightly complicated solution-based mechanism. I think the most important thing to bear in mind is that in solution, you do require both the nitrite in the protonated form, so nitrous acid form, but you also need the amine in the deprotonated neutral form as well. So in some ways they pull in opposite directions, as this next slide will show. That if you look at the top right at the moment, the top right, the plot there, what you see is the amount of species in a non-ionized form. So as you go to very acidic conditions, the nitrite becomes in the favorable nitrous acid form. So that is favorable for nitrosamine formation. But on the other hand, you also need the amine to be in its deprotonated form. So higher pH is the favorable conditions for nitrosamine formation. And so it's only where you have an overlap, really, that you get nitrosamine formation really being very rapid. And really, that is really summarized in the contour map, really, in the bottom right, which is showing how the combination of having an amine with a low pKa in combination with a solution that is acidic are the worst conditions, that i.e. nitrosamines are formed most readily with a low pKa amine and in an acidic solution. But that's all in solution, but that also serves as very good background for trying to understand what's going on in a solid. So the next thing I'd like to just talk about in general is how common nitrites are in excipients. Yeah, as I said earlier, that it's, nitrites haven't historically been mentioned in C of A's, even though they're there at ppm levels. They are found in ppm levels in many excipients, and I'd just like to give a little plug to the external database on nitrites that is being collated by LASA, and no doubt that's been already talked about in previous presentations. And we just see here a snippet of the sort of excipients with their nitrite levels. I'll come on to that a little bit more later. But really, I'd like to talk mainly around first of all around nitrites, because understanding the chemistry of nitrites is half the battle, really, in terms of understanding nitrosamine formation, because nitrites are a precursor to nitrosamines, which is the obvious point. But the first important thing about nitrites is that they are volatile, really. Their levels can reduce over time. So what this slide shows is, on the left, we had some preliminary lab scale data that every time we opened the container and tested this was microcrystalline cellulose. Every time we tested for nitrite, the levels seemed to go down. And we did a specific laboratory scale experiment to look into this because we wanted to look into the effects of temperature and humidity on the rate of that process. And sure enough, we got very trendable results when we looked specifically at in different temperatures and humidities on the rate of loss of nitrite. And then to confirm all of this as well, we also compared open versus closed containers and saw quite a difference. So this sort of explains why nitrite levels can remain reasonably high on storage if the package is kept untampered with, if you will, and the lid on. So nitrite levels could remain for a very long time, but as you start to reduce the bulk scale, because as things get smaller and smaller, there is more surface area, etc., and the more you open and close the lid, the nitrite levels do tend to go down. And we scaled this up. We did a larger scale study into this, because obviously this is an opportunity in many ways, to, you know, can we in some way condition the excipients to get lower levels? And sure enough, yes, even at scale. So this is literally blowing air through a powder to get the nitrite levels down. And we got a very nice trendable data there. And as you can see, it is possible to even develop a model for the rate of loss of nitrite based on temperature and humidity. And that's what we did. But and then so we looked into this a little bit further. And actually, before we did the experiment on the right, we thought, well, let's turn the temperature right up. Let's look at 90 degrees, just to see. That should really get rid of the nitrite very, very rapidly. But strangely, that didn't have, it didn't reduce the nitrite levels as much as we'd hoped, actually. They stayed pretty constant. And it was only when we did the fuller experiment on the right, where we looked at temperature and humidity, that really perhaps there was an explanation for why 90 degrees didn't work, in that it's a very dry condition. You know, the humidity would have been less than 5%. So really, this is all pointing to the key role humidity has in almost liberating that nitrite. It makes it into a volatile state. And as we'll see later, not only a volatile state, but a more reactive state as well. And, you know, for people who are interested in modeling and the effects of humidity, which I know FreeThink are certainly interested in that, it's sort of not really that surprising that the relative humidity, if you really want to model this, then look at the relative humidity rather than the absolute humidity to really start understanding what that relationship is. But I don't really have much more time to talk about that. I need to move on. And so, coupled with that observation that humidity and the nitrite levels drop over time, I'd like to just draw attention to this very, very simplistic experiment, really. So what we have here are 4 scenarios. We have sealed and open, and we have a situation where an API was mixed with an excipient versus where the API was physically separated from the excipient. So hopefully you can see that, the 4 different scenarios. Now, what we found was that when you physically mix the API with the excipient and put the lid on, I suppose it's not that surprising that the nitrosamine level in that scenario was the highest. It was, you know, 147 ppm, for example. Now, when you did the same experiment but keep the lid off, now the level of nitrosamine formed was significantly less. It's, as you can see, 25.6 ppm. So that's really emphasizing how packaging is going to be key here. And also, you know, and it really underlines the importance of volatile reactive intermediates in the nitrosamine mechanism. But then, interestingly, also on the right-hand side, where we see the physically separated material, if you look at the top right scenario, we get 7.6 ppm. Now, we interpret that as that is significant. Even though it's lower, it's still significant, and it's a very important observation, because even though the API is physically separated from the microcrystalline cellulose, the microcrystalline cellulose was still able to bring about the nitrization through the headspace. So that really shows that the same species that are being lost when we blow air through an excipient and making the nitrite levels come down, those same species also have a role to play in actually the nitrization reaction with an API. And so finally, the bottom right just serves as a control, really, for this. It's open and it's separate, and not surprisingly, we got lower nitrosamine levels. So that's a very significant observation, even though it was a very simple experiment, it does tell us a lot about the nature of the nitrosating species. And in a sense, my co-workers, Greg Sluggett and Todd Zaleski, actually, they had a told you so moment, really, I guess, with this, because back in 2018, they published a paper that was focusing on how to achieve 60% RH. That was the focus, really. And one of the saturated salt solutions that can achieve 60% RH is sodium nitrite. And they discovered that they got artifactual formation of nitrosamines with 3 APIs, secondary amine APIs, or at least some of these, carbodiol, propanolol, and norfloxacin, which are all secondary amines. Some of those showed very significant nitrosamine formation, which was at the time put down to volatile nitrosating species in the desiccant, in the desiccator, sorry. Yeah, and we've also seen examples of API forming nitrosamine when dried in an oven containing excipient with high nitrite. So this is the NOx, if you will, the nitrous oxide gases are something very important to be aware of when talking about nitrosamines. And then just finally, this was a study that was conducted in Chennai, where the air quality isn't sometimes the best. And this was one of our more reactive APIs, sat on an open bench. But as you can see there, as that API sits on the open bench with, literally on its own, the nitrosamine levels did increase as seen there over time. And I've just drawn a sort of diffusional curve through that just to link those dots, which is one of the fit models that is used in ASAP Prime, for example. But so that is showing that just an API on the open bench can pick up nitrosamine. Some APIs can certainly do that. So this all brings us on to mechanisms, really. What can we understand from all of these observations? Well, putting it all together to some extent. So on the left, we have the excipient environment, if you will. So it could be nitrite, as an anion. It could be protonated. It could be nitrous acid. The fact that it's volatile might indicate that it is nitrous acid. Because nitrous acid does have a significant vapor pressure. But also other types of species has been talked about, different species of nitrosating species, such as nitrite esters. They're theoretically, very likely, except I must admit, we haven't got direct evidence that such things are formed, but nevertheless, if they are there, they could be nitrosating species in themselves. It doesn't necessarily have to go via nitrous acid in order to form nitrosamines. There is a direct route across the bottom there. So I'm painting this picture where we have the excipient environment on the left, and in the center of all of this, piecing together the evidence, we have a nitrogen species, probably in the third oxidation state, nitrogen 3, that does play a role in forming nitrosamines. We can see that we've pieced that together. We've seen the loss of nitrite. We've seen the through space nitrization. So I think that is a very significant contributor to the mechanism, these gaseous species. And there are many things it could be. It could be N2O3. It could be HNO2. It could be the combination of NO and NO2. All of these things are in a state of flux with each other. So I don't currently yet get too hung up about tying down which one of these is truly responsible, because they are going to be somewhat in a state of flux with each other. And this explains how there is almost now a competitive process on the right. Once you form this reactive volatile species, it can either be lost to the environment or it can form nitrosamines. So I'm setting this up as a competitive reaction, really, which sort of explains our observations with the systems that we've studied. So I'd just like to say a little bit more on this whole state of flux, if you will, because there is a lot of debate around what is the species, what is the nitrosating species. Now, I must admit, I'm fairly relaxed about that question because as I say, they all interconvert. So it becomes a little bit of a moot point about which one specifically is responsible. I know it'd be nice to tie down a detailed mechanism, but ultimately the effect is the same. And analytically speaking as well, that's what this slide is talking about. Analytically, this is trying to make the point that they would all manifest as nitrite because, for example, nitrite esters in an aqueous solution are likely to, the position of equilibrium is likely to be pushed very much towards the nitrite end of the scale. So it will be manifest as nitrite. And similarly, when we do things like, so depending on the analytical method, if it's ion chromatography, for example, it's often the direct measurement of nitrite, which is fine in an aqueous environment and in a pH that's 4 or above. The majority of all of these nitrosating species will be manifest as nitrite. Similarly, you might be using a derivatization method, such as the Dan reagent, for example. But similarly, that again, the same is true there, that all of these species in this cloud that's drawn on this picture will all nitrosate Dan, and so they will all manifest as picking up on the Dan method, if people are familiar with the Dan method. So, I'm personally quite relaxed about what is the specific nitrosating species, but then I'm being quite superficial with that, but I'll move on. So competing reactions, we believe this competitive reaction scenario explains many of our observations. So for example, when we did the experiment open versus closed, we got higher nitrization in the closed scenario. So immediately that points to this competitive situation where the nitrosating species can either be lost to the environment or nitrosate. And by opening the lid, you're just making the loss to the environment so much quicker. And what we often find is the other thing, as a slight aside to this, is that we do see plateauing behavior. Now, that's not surprising. That's not surprising at all, because we do have a finite source of the limiting reagent, which is typically nitrite because it's present in ppm levels. So, it's not surprising that you get a plateau level formed, which could correspond to 100% conversion of the nitrite into nitrosating species, but not necessarily 100% conversion. Come on to that later. And what we found was that, when you increase the rate, you increase the plateau level. So as drawn here, so the steeper the onset, the higher the plateau level. And so that is all consistent with this competitive reaction. So coming on now to the effect of temperature and humidity, we've already talked about the importance of humidity to drive the loss of nitrite. And so this is a slide where just one by one you say, what is the effect of temperature? What would temperature do? Well, of course, temperature makes everything quicker. Everything quicker, really. It would, even, physical processes like the loss are going to be more accelerated through higher temperature. And all reactions, all chemical reactions are going to be quicker at higher temperature because of the Arrhenius equation, for example. So temperature makes everything quicker. Temperature accelerates the whole process. And in a sense, in the systems we've studied, now I don't claim that this is true for every system, but what we did, we looked at, when you look at different temperatures, what is the effect? So what we found was, because temperature makes everything quicker, even the loss, then the affects the rate, but in this case, the plateau level remains the same. So we were encouraged by that, because we were saying, well, if we want to understand what goes on at 25 degrees, then what goes on at 60 degrees is representative of what will go on at 25 degrees, but much quicker. Because the important thing for us was we need to know what that plateau level is, really, because this is a fast reaction, even in the solid state. It's likely that that plateau level in many, many systems will be reached within 1 to 2 years at room temperature. So for us, with our products, it was all about understanding that plateau level. It wasn't really about understanding the kinetics, if you will. Next, humidity. Now, we've already seen the effect humidity has. It really drives it to that volatile species. It really does drive, we know that from the way nitrite is lost under high humidity. So we do know that humidity really pushes it to that reactive species in the middle. But then humidity won't really have much of a role in accelerating the loss to the environment once you've formed that reactive species. So this is consistent with our observations again, that humidity affects both sort of the plateau level and the rate, if you will. So if you can see the difference between the top plot and the bottom plot, humidity, it does have a role in affecting that plateau level. It's almost like higher humidity increases the amount of available nitrite, really. That's what we seem to observe. And in fact, we took that a step further with one of our products. I know the writing is very small on the left, but what they represent is a number of different packaging types where the water activity was measured. And the water activity is very synonymous, really, with relative humidity. And so when you try and correlate now, the different nitrosamine levels, and in this plot, all of these data points, we believe, had reached their plateau level, if you will. So what we see now is a correlation between the nitrosamine plateau level and humidity. And this is what we saw with one of our products, that you see this sort of gentle curve, and the line drawn through that is exponential, which again is totally the way ASAP prime is, the assumptions in ASAP prime that the reaction rate increases exponentially with relative humidity. Well, there does seem to be many parallels here with the way humidity affects the nitrosamine plateau level. And so I've taken that curve on the left and then talked about conversion of nitrite, because of course, in those systems on the left, we know how much nitrite was present in each of those data points. And so we're able to convert that curve now and express it as percent conversion of nitrite as a function of water activity. And I guess that's a pretty definitive observation for this system. I won't go into much more in terms of trying to explain that or understand that, but it is a pretty solid observation that we saw. So moving on, that sort of leads slightly into ASAP. We've already had a hint of the way perhaps ASAP could be useful, because in the systems we studied, we got the same plateau level at 60 degrees as we did at 40 and 25, etc. So ASAP was a very good way of very quickly getting a measure on the plateau level. But what there is a few cautions here. First of all, we found that we needed to keep the lid on, because of course a lot of ASAP studies use humidity control ovens where it's necessary to keep the containers open to get that humidity equilibration. If you do that, you do run the risk of getting falsely low data, certainly with the systems that we study. So all our ASAP studies really were closed systems, which really limits us, really, because we're not able to do that much in terms of humidity control. Because we were also worried that if you did use a salt solution in a closed environment, that the salt solution could potentially, particularly if it's alkaline in nature, act as a sink for all those nitrosating species. And so, we had slight concerns of having quite large quantities of salt solution in a closed vessel with very tiny amounts of nitrite that we're trying to understand. So we've shied away from using salt solutions. And we tend to do a lot of our ASAP, well, you wouldn't really call them ASAP studies anymore, but we do them with natural humidity. But we are mindful of the fact that humidity can affect our degree of conversion, as seen with the previous case study. So yes, in the interest of time, I think I'll move on actually, because I am conscious that time is ticking on. But we did, occasionally we do get a chance to do a thorough long-term versus ASAP study. And the data set that we have at our disposal was one where we looked at different batches that do contain antioxidant, which I'll come on to later. But we saw a very good correlation between ASAP, the plateau level after 2 weeks at 60 degrees versus the plateau level that was observed after, 12 months storage at 25 degrees. Now, what you can see is there is a slight bias, if you will. So theoretically, if it was a perfect prediction, all of those dots would lie on that diagonal line. Now, what we see is ASAP slightly over predicted the amount. So the plateau level at 60 degrees was slightly higher than the plateau level that has been so far observed at 25 degrees. Now, I think that is a very good way around for a start. First of all, it's showing that ASAP is a cautious estimate of, the nitrosamine risk. So that is good. It's the right way round. But not only that, there is an explanation in that the ASAP containers are less permeable than HDP bottles. They have a tighter head. We deliberately chose a very small headspace configuration. So the losses were slightly less in our ASAP conditions than the losses that you might see in an HDP bottle with a big headspace and with permeable plastic. And not only that, but there is a risk, there is a possibility, shall we say, that the 25-degree data isn't plateaued fully yet anyway. It's still got a little bit of growth to go. So for me, those are good enough reasons to say why ASAP slightly over predicts. But on the whole, I was very, very pleased to see that this correlation across different batches. So coming on to API reactivity, we all know that, as we said, pKa is an important structural consideration. So is the aryl versus alkyl amines, etc., etc. So I'd like to, talk about solids, of course, API reactivity in solids. So in solids, the API reactivity will affect both the rate and extent of formation. That is to say, because we have that competitive mechanism that's drawn on the screen again, that the faster you make that lower pathway, if your API is particularly reactive, that pathway obviously becomes more prominent, and therefore the rate obviously gets steeper on the plot. But because your more of the nitrite goes in that lower pathway, that affects the plateau level. So we'd expect both the plateau level and the rate of formation to be affected by the API reactivity in solids. And the other point to make is in solids, it's not just about pKa and the structure, the chemical structure. There are other considerations like particle size, surface area, crystal, crystallinity, degree of disorder. All of those factors that affect solid state reactivity also will now start to have an effect on API reactivity in the solid state. But so this really led us to, based on our understanding so far, we thought, well, there are some very good industry standard tests that you can do. There's the NAP test, which is all done in solution, and we have IQ conditions, which are built to really test if an API is susceptible to nitrosamine formation in solution. But we thought, well, given that there are additional considerations for solids like solid form, choice of salt, etc., really all of those factors are invisible to the any solution-based test. So if you wanted a way of discriminating between a reactive API crystal form versus a non-reactive API crystal form, then the NAP test isn't going to pick that up, because it all dissolves, it all goes into solution, and they all behave the same regardless of salt form, counter ion, all of those considerations, because it's only the pH of solution really that would have any effect there. So as you can see here, we came up with this way of screening different APIs for the solid state reactivity. As you can see, there is a physical separation between the nitrite and the API. So we're talking about powders here, and we're relying on that through space nitrization to really get a grip on the API reactivity, which we think is very, very relevant to understanding the nitrization risk in solid drug products. And as you can see here, we did see a significant difference between different salts of the same API. These are of the same API. Interestingly, and perhaps counterintuitively to some extent, the free base of this API was far more reactive than the two salts that we looked at. So on the x-axis, we have the rate of nitrosamine formation in this screen that's drawn to the left there. And on the y-axis, we had the amount of nitrosamine formed in a real-world scenario, which was a binary mixture of our API in MCC. So this was a very good way of getting a grip on how reactive this API was in the real solid drug product. It's quite a good correlation. More work needs to be done, of course, on this, but we was very pleased with that correlation. Which then now brings us on to formulations investigations, again, carrying on from that previous slide in a way. So this is a plot that's showing how different nitrite levels in formulation correlate to the amount of nitrosamine that is formed. So again, the y-axis is really that plateau level. How high does the nitrosamine level actually get? Interestingly, those dots are what they are. Each dot represents a different excipient. Again, I'm not really attempting to explain this other than this is what we saw. But what we do see is different degrees of conversion. So for each of these excipients, we did measure the nitrite, and we did measure the amount of nitrosamine formed. And, as you can see, it wasn't 100% conversion. The correlation, I suppose, was 0.79, as it says there. And of course, that does show that understanding the nitrite levels is important to assess the risk for solids, but it's not the only story, really. And the time doesn't really allow, because we looked at multiple different APIs, free bases, salts, etc. All of that does play into the conversion, the percent conversion. That all, the choice of salt does affect the conversion, as we saw previously. Obviously, each of these excipients have different humidity levels, which again is another area. It's not that surprising, therefore, that perhaps we do see a range of different conversions, because each excipient does have a naturally different relative humidity. And so, that isn't really something we've corrected for in this plot. And then just finally, this isn't really that important, but what the previous slide showed was some binary mixtures. They were all API excipient binary mixtures. And we was curious to know, well, if you know the way binary mixtures work, does that allow you to work out how a multi-component mixture would work? And so sometimes they behave as expected. So in this case, where we've got lactose as an individual excipient, and that was the level of nitrization we saw with lactose, and it had about 1.7 ppm of nitrite in that lactose. And then we mixed that with MCC, which you can see there as the blue sort of dot. The mixture of the two should, if those two excipients sort of act independently on the API, if you will, without any sort of interaction parameters, then you would expect the mixture of a 2-to-1, as you can see here, it's 64% MCC, 32% lactose with a few other ingredients. That should probably lie 1/3 along the way between the MCC and the lactose if there were no multi-component interactions going on. And by and large, it does. Within experimental variability, that does lie between those two individual components. But then we do see cases whereby we typically see higher levels in the multi-component mixture than would have been predicted on the basis of binary mixtures. So this plot on the right shows like an excipient called ProSolve, and we mix that with lactose. So we have the data from the binary mixtures initially, but then we have data from a multi-component mixture, and that multi-component mixture had higher levels of nitrosamine than might have been expected just on a sort of basic, like linear combination of the two ingoing excipients. So that remains an area of focus, really. Certain excipients, certain blends of excipients have an interaction that sort of have an effect beyond the way those excipients would have behaved in a binary mixture. And that could be down to things like localized pH, for example. Some excipients, even though they don't bring any nitrite, they change the pH environment, for example. That's a possibility. They could have different water activities, as we mentioned already. We somewhat suspect the ones that seem to have this activating effect, i.e., when you include them in the formulation, they seem to bring about a higher conversion level, even though they don't bring nitrite themselves. They tend to be ionic in nature. And so that's what we've observed. So it could be something that's they might be interacting somewhat with the API salt, perhaps, for example. So anyway, yeah, moving on. These are my last few slides, so I will stop. So high pH is often talked about as a strategy for controlling nitrosamines. Now, that is certainly true for a solution, but we tested this with a solid, actually, and we took an extreme example. We took an API-free base, so there was no acid counter ions because we wanted to really get a high pH formulation. And we used magnesium carbonate, which is one of the most alkaline excipients out there. Now, unfortunately, magnesium carbonate also has a huge level of nitrite in it. But nevertheless, even in this very, very alkaline environment, we still got 40% conversion of the nitrite into nitrosamine. So with the experiments that we've been doing, we didn't be able to really confirm that controlling pH in a solid is a good strategy for controlling nitrosamines. So that's one thing that we did conclude from the data so far. I know that is potentially a controversial point. So next, I'm really summarizing now. I haven't talked about antioxidants really, but I'd just like to recap a few of the things I've talked about, but also just briefly talking about antioxidants. So antioxidants or inhibitors can be an effective strategy. The FDA mentioned that as a useful strategy that we should look into. And we did see varying degrees of success with different antioxidants. So this is API-A, as you can see, adding sodium ascorbate, we got a modest improvement in the nitrosamine, nothing to write home about there. Propyl gallate, better still, but still not brilliant. And then cysteine was quite good. So cysteine, yeah. But then for API-B, we saw that propyl gallate was extremely effective. We don't understand why particularly we see this difference. in terms of why different antioxidants are better than others for different APIs. And then finally, with API-B, we've looked into how much do you need to add. Now, it's not that surprising that the more antioxidant you add, the lower the nitrosamine formed. It's not a simple cutoff. It's a curve. You can fit a curve to this. You could argue it's diminishing returns. The more antioxidant you add, the less benefit you get. So the first few percent of antioxidant added has a very significant reduction, but then you get diminishing returns. And so just to, it won't be surprising to know that we've modeled this, because what you have almost is you may have remembered from presentations I've given that I talk about a contact surface area sort of model. And that would account for these sorts of observations, that if you consider what is next to what, is the API next to excipient, is it next to antioxidant, etc. Now, all of that has to be somewhat countered by the fact that we know that these species are very mobile and volatile. So for this, I don't talk about contact surface area, I talk about proximity. So it is possible to model what we observe here in terms of a proximity model, which again, we don't have time to go into, but that's an interesting area of development. So in summary, this is my last slide. I think we made quite significant progress in understanding mechanisms that are relevant to the solid state. The relationship between nitrite and nitrosamine. Not all amines nitrosate, and nitrization tests have an important, like the solid state screening tool that I outlined earlier, could have a very important role in understanding solid state reactivity. I think I've really emphasized the importance of the volatility and mobility of the reactive nitrosating species, really to understand solid state nitrization. And I've also talked briefly about some of the factors that can affect that percent conversion. So the intrinsic API reactivity, the drug load, we didn't have time to go into that very much, humidity, temperature, and these activating excipients, for example. And I think it is sort of emerging that there is a difference between solid state mechanisms and solution state mechanisms. I think for me personally, I wouldn't rely too heavily on solution state kinetics to really understand solids personally, but that's a sort of personal view. And yeah, I've talked briefly about nitrosamine reduction strategies. Obviously low nitrite excipients, we've seen that. We implied, I didn't talk about this directly, but of course the humidity effect. Desiccants are key. They can be key for controlling nitrosamine levels, that percent conversion. Antioxidants, we all know about that, but antioxidants do come with a lot of baggage, discoloration. They are by nature reactive. And so you have to be very careful before you put too much of a reactive excipient, if you will, into your formulation. It does come with a lot of baggage, and it is a reformulation. And different salt forms. I think we're forming an understanding of what would make a more protected sort of salt form. And we've also looked at processes for reducing nitrite levels and excipients, like that conditioning experiment that I talked about at the sort of beginning of my slides. So with that, yeah, that is it. Thank you for your attention. And yeah, I'm really sorry. As usual, I think I went through a lot of things very quickly there, but feel free to reach out. We have. I've found my diary has changed significantly recently, so yeah, I will try and make every effort to connect on this topic. So thank you.
Thank you, Gary. There are quite a few questions, and we'll try to get to at least a couple of them that I think are worthwhile. Let me ask one of the questions that was asked, and something we've observed is in many cases the nitroso species that you form itself degrades over time. Have you seen, so you talk about plateau, in the examples you gave, did things also then decrease, or was it always just flat?
Yeah, I think that is a very API related phenomenon, actually. So our APIs, once they form a nitrosamine, they do appear to keep it. I think they don't go on to react that much. So once it's
So your examples were all where the N-nitrosamine was on the drug, rather than the small molecule ones like the dimethyl N-nitrosamine.
yes. Oh yeah. Well, the ones I talk about are on the drug as well, actually. So
No, that's what I'm saying. Yours were on the drug
yeah.
versus other ones that are small.
Oh, of course. I mean, that is absolutely true. If you've got a nitrosamine which is in itself volatile, then
Well,
that's
whether
going to,
it's volatile or could be reactive, depends.
yeah, absolutely, totally, yeah. And also there's a lot of things that we don't think of as volatile, but they still have a vapor pressure as such and can start to
Yep.
really affect, the measurements we make. Unfortunately, yeah.
All right. Another question. You mentioned that these nitrites that are precursors could be volatile under the right conditions. If you were to treat excipients by, let's say, fluid bed treatment, would you be able to remove enough of it to matter?
Absolutely. Definitely. I mean, that's what my slide showed, actually. You're going back to the top of the slides. That's literally what we did.
But did you do your fluid bed with some kind of humidifying?
Yes. So,
Okay.
yes. Now, that is
So it's
difficult.
not a normal way you would run a fluid
Yeah,
bed dryer.
absolutely.
Normally you run them dry. You're saying you try to find a way to use humidified air as part of your drying process or part of your fluid
Yeah,
bed process.
I understand they really had to challenge the setup to literally turn the tap of it's literally a tap of water that they had to turn up to the very max in order to get those reasonably humid conditions. Actually, well, they're not actually even that humid, by our standards. It's 25, 50. That was it's so so in order to get that 50% RH, they had to really turn the tap of water up full and get
Right,
that.
but if you could dry it at, let's, dry at least the placebo part of the blend at, 50 degrees, 80% humidity or 75% humidity, you might
Mm.
be able to do even better.
Oh,
Okay.
absolutely, absolutely, yeah, that's right.
Okay,
Mm.
another question, one of the ones dear to my heart, but I'll throw it to you, is you mentioned that water activity is a better measurement than water content. And why would you say that?
Well, I mean, first of all, the evidence itself, rather before getting, so we do whenever we use water activity instead of water content, we tend to see much better trends with whatever stability phenomenon we're trying to understand, be it, rate of chemical degradation, or in this case, the percent conversion of nitrosamine formed, percent conversion of nitrite, sorry. So now water content is so, there are so many reasons why that is often a bad measure, because not least because of the analytical variability, very often the data isn't very reliable for that reason. Also, there is a lot of things like water of crystallization that is taken up into, and that won't be playing a role in reactions really. It's tucked away where it's not doing anything, and yet it still contributes to the water content. So consistently, time and time again, if we're looking to monitor and understand the stability related phenomenon, it's always water activity that is makes much more sense when you trend it against that stability phenomenon that you're trying to understand, than water content. Water activity is definitely the one to go with. But yeah, I mean, and I don't want to get too into the theoretical reasons for that, because it's not that straightforward to make a theoretical argument for it. But certainly The
I think, yeah,
you do that, yeah, absolutely. I wouldn't be doing that now for me now where I'm sat because I'd need to think about it. But certainly the experimental observation is that, yeah, yeah.
all right. I'm afraid we're out of time. I had more questions related to how you define base and solid state, acid base, but I think we'll leave that discussion for another time. And appreciate your talk. And anybody having further questions can reach out either through FreeThink or directly to Gary. And we'll try to get answers to everything. These slides, as well as the recording, are going to be available relatively shortly on the FreeThink website under webinars. Thank you, everybody. And this concludes our webinar series for this time. Be on the lookout. We'll have another series coming up next quarter. Thank you.
Thank
Bye,
you very much.
everybody.
Bye-bye.